So just to give you a brief background of myself, back in the day I used to be a professor at the University of Ottawa, and I was a scientist with the National Research Council of Canada, doing a lot of work in immunogenetics and cancer immunotherapy. And I think if anything more so, these days, you need to have a lot better understanding of immunogenetics considering what's going on in the global scale, especially regarding coronavirus. There is a chunk of susceptible population out there that does succumb to the virus, and that's really your underlying immunogenetics. It's about how your body is mounting an immune response to the virus. If the immune response is substantial enough that it cannot be controlled, then you will in fact succumb to the disease. It's not the virus killing you, it's your immune system at the end of the day. But clearly with coronavirus, we don't have availability of that data, and it's definitely going to be a while before we get our hands on that data. But if we could understand the underlying immunogenetics, you can almost predict who's likely to be more susceptible. It's not as simple as saying that someone is in their 60s or 70s, and just because they're that old, they're susceptible. From an immunology and a genetic standpoint, that does not mean anything. It's about what's really going on in your body. And same actually goes for when it comes to nutrition, detoxification, breakdown of hormones, and almost everything that's in your body, because your DNA is the pathway that your body works on. And today our focus is going to be on the genetics of nutrition and detoxification. So, as I was saying earlier, perhaps the biggest discovery in medicine was the mapping of human genome project. It took around 11 years to complete, but again, after those 11 years, what you had was a big code that is pretty much a lot of ATGCs, and that's about it. But that's where we really opened the door to personalized healthcare. So when we talk about personalized healthcare, we're really talking about giving, whether it's nutritional guidelines, fitness guidelines, medication, or anything that you talk about based on individuals' DNA. For instance, nutrition. Because we have underlying differences in our DNA, some people will have very unique nutritional requirements, and that's where you have to cater it based on their DNA. Similarly, fitness. Certain exercise regimens are going to be having a lot more efficacy for certain individuals versus others, and that's in the DNA. The other aspect is pharmacogenetics. So with any medication out there, it only works on around 40 to 45% of the population. And for a big chunk out there, there is no effect of that medication. But if you were to understand the genetic makeup of an individual, you can identify what their response is going to be to any medication out there. If they'll have an adverse reaction, if they will see a strong positive response to that medication, or no response whatsoever, and that's pharmacogenetics. The other one is, of course, disease risk. For instance, BRCA1, BRCA2, those genes are associated with breast cancer. Similarly, that's the case for a lot of diseases out there, with a lot of cancers. The aggressiveness of a cancer has an underlying coding in your DNA. Because in our body there are genes that are called tumor suppressor genes, and their function is to just suppress tumor formation. But imagine if you have a mutation in one of those genes, then tumor formation in your body is not suppressed, and that can expedite generation of tumors and hereby cancers in your body. And that's what we say when we talk about disease risk based on genetics. So DNA, quite simply, we can refer to it as the molecule of life. It stands for deoxyribonucleic acid. And all of the eukaryotic cells, which are cells excluding bacterial cells, they have a big structure in the center which we refer to as nucleus, and within the nucleus you have chromosomes. So chromosomes are essentially bundled DNA which are wrapped around into proteins called histones, so it's a very tightly packed structure. And as humans we have 46 chromosomes, 23 chromosomes coming from the mother and the other 23 from the father. If we look at the structure of DNA, it's composed of nucleotides, and those are what you call ATGC. But overall, looking at the molecular structure, of course you have a sugar backbone, which is bound by phosphodiester bonds, and then you have these individual nucleotides, which is ATGC, and so on and so forth. And when you look at three of them together, that is referred to as a codon. And one codon codes for one amino acid, and that's how you get the hierarchy of DNA, then RNA, and then protein. What is a gene? So again, that's a chromosome, and a chromosome, as I said, is really bundled up DNA. Part of the DNA that codes for a protein is one gene. And so far, around 30,000 genes have been identified in the human genome. So what is the hierarchy when you talk about going from DNA to protein? So you have your DNA in the nucleus. From a DNA, you get a messenger nucleotide form, which is referred to as RNA, and this is the recipe for generating a protein, whereby every codon or every 3 nucleotides code for a specific amino acid. And as you can imagine, there are multiple codons on here. And for every codon, you get an amino acid, those amino acids tied together, and you get the protein. So how do mutations play a role in impacting enzymatic activity or the functionality of a protein? So let's say this is the normal version of a gene. So with the gene, you get a functional enzyme. And the function of this enzyme is, let's say, to take this circular substrate and to turn it into a square form. And the reason why it can do it is because it has a certain 3-dimensional cleft, which has a high affinity for the circular molecule. Now, let's say, hypothetically, somebody has a mutation in the codon that codes for a part of the cleft. It's going to be a single nucleotide chain, but what it can lead to is a complete loss of function, because now all of a sudden, this substrate cannot bind into the cleft. And because you have no binding into the cleft, you have a mutated enzyme where you have no activity of the enzyme at all. And that's how a single nucleotide chain can have a dramatic effect when it comes to enzymatic or protein activity. So coming back to nutrients. So overall, when we talk about nutrient absorption in cells, it's really a 2-step process. First of all, you have proteins or enzymes, which are nutrient transporters, and their function is to take the nutrient into the cell. And with a lot of nutrients, it's really a 2-step process whereby you have another enzyme present within the cell, which is going to bind to the form of the nutrient which is coming in, and it's going to modify it to a form which is bioactive. In other words, that can be used by the cell. So a quick question, what do you think happens if there is a mutation in this protein? What's going to happen?
Nutrient might not make it in.
Exactly. So imagine somebody is carrying a mutation in this transporter for any vitamin. You can be giving them as much as you want of a specific nutrient, but it's not going to be going into the cell, and they'll still have deficiency. On the flip side, let's say somebody has a mutation in this enzyme. What happens then?
Cannot make it bioactive.
Exactly. So for those individuals, what you need to supply is really the bioactive form to make sure they do not develop deficiency. And this is really a very simplified schematic to illustrate how DNA plays a role in nutritional genetics. So when it comes to nutrients where there is a clear association with the underlying DNA of the individual, there are of course a lot of them. We won't be covering all of them today, but the common ones are vitamin A, B6, 12, choline, C, D, E, and so on and so forth. But to illustrate the point, I'm actually going to focus on 2 examples. One is vitamin A and the other one is folic acid. So when it comes to vitamin A, there are 2 main sources of vitamin A. One is from animals, which is retinol, and the other one is, of course, from plant-based sources, which is beta-carotene. But at the end of the day, it's retinoic acid which is being used by your body. But for beta-carotene to be converted to retinoic acid, there is an enzyme called BCO1 which converts it into a retinaldehyde first. And it turns out a lot of people do have a mutation in BCO1. In other words, in these individuals, you can give them a lot of beta-carotene, but they'll still have deficiency for vitamin A. If anything, it's almost 45% of the population that has this mutation.
Wow.
So they have an impairment in activity of BCO1, and the specific variant is really called the T allele variant, and that results in 69% reduction in enzymatic activity. So let's say if someone has the T variant of BCO1, how would you deal with that? What would you do?
To give them retinol.
Exactly. And that's something which you can only identify once you look at their underlying DNA. The next example we'll briefly touch is folate. So of course, you guys know it's an essential nutrient. Deficiency can lead to anemia, and in pregnant females, that can lead to neural brain defects in babies. Now for folic acid or folate to be converted into a bioactive form, one of the key enzymes is MTHFR. So MTHFR really converts folic acid into 5-methylfolate, and that is the active form which can be used by the body. But of course, as it turns out, a lot of people have mutations in MTHFR. So again, I won't be going into specific mutations, but the take-home message is that around 30% of the population is in fact carrying a mutation in MTHFR, which can reduce the activity of the enzyme down to 25%. So for these individuals, you can give them all the folic acid there is in the world, they'll still have deficiency. You need to supply them with 5-methylfolate. And again, this is something that you can only understand as when you look at the underlying DNA of the individual. So just to summarize, so what does nutritional genetics, what is it that we get out of it? So using genetics, we can identify for which nutrients individuals have a higher risk of developing deficiency to. And at the same time, we can develop nutritional plans that takes into account all the information and really identifies the right source of nutrition for that individual to make sure they do not develop deficiencies. But the biggest message is, what this is telling you is that you cannot have a generic nutrition plan for everyone, because people have those underlying differences. And you have to factor in those differences to make sure you can have efficacy such that those individuals don't develop those deficiencies. The next aspect we'll briefly cover is weight loss in genetics. So usually when you guys come up with a dietary plan for weight loss, there tend to be 3 different outcomes. For some individuals, there will not be any weight loss. Some will perhaps see a little bit of weight loss, and some individuals will have a dramatic weight loss. Why is that the case? Why can't it just be dramatic weight loss? Again, the reason is the underlying genetics. And I'll illustrate the point using 2 different examples, one from a dietary standpoint and one from a fitness standpoint. So monounsaturated fats. Of course, you look at newspapers, it's going to say you have a lot of monounsaturated fats and you'll lose a lot of weight. Yes, that is the case, but that's only for a small chunk of population that have a certain genetic makeup. For the rest, having a lot of monounsaturated fats, it's not going to help them lose weight. And the specific gene that plays a role in the process is PPAR gamma 2. So this is taken from a study which was conducted, I think, almost 10 years ago. And the real take-home message is, so there are 2 versions of this enzyme. So let's just say one is the C version and one is G version. Individuals that are carrying the C version of this gene, even if you give them a diet which has over 56% monounsaturated fats, they do not lose weight. But individuals who are carrying the G allele, if you were to give them a diet which is high in monounsaturated fats, you see a significant weight loss. Again, it brings it back to the same point. You have to understand the genetic makeup of the individual to identify what their body needs, even when it comes to weight loss. Now, the other side to weight loss, in addition to diet, is fitness. And again, the common thought is, you do a lot of strength training, you gain a lot of muscle, you'll have increased calorie consumption, and of course, you lose weight. Yes, that is the case, but again, strength training does not work for everyone. For some people, doing a lot of strength training will actually lead to increase in fat mass and not muscle mass. And that's where a gene called INSIC2 comes into play. So again, when it comes to INSIC2, there are 2 different variants of the gene. One is the G variant and one is the C variant. So individuals that were carrying the C variant of the gene, when they were doing a lot of strength training on their biceps, they had a significant gain in fat volume, not muscle volume. In contrast, individual who had the G allele, that was not the case. So again, you look at someone's DNA, and if you identify that the individual is carrying C allele, you should not be recommending them to do a lot of strength exercise, because if anything, that will lead them to gain fat and not gain a lot of muscle. So this schematic is taken from Wall Street Journal from around March 2010. And the point that they were trying to make was that overall, if you come up with a dietary plan for an individual, which is generic, the median loss in weight is around 4.6 pounds. But if you were to prescribe them a genotype or a DNA-based diet, it's around 13.2 pounds. So around 3 times more. But again, the real message that I want to bring forth here is that if you can prescribe somebody with a DNA-based weight loss plan, which includes both diet and fitness, then you're increasing your chances to have dramatic weight loss in that individual. And yeah.
How many different genetic markers are you able to categorize for weight loss?
So it's around, what we look at is over 35 genes.
Wow.
Yeah. This is, again, a very simplistic version.
Yeah.
And because there are so many different parts when it comes to genetics, I'm just illustrating the simplest examples of every single topic. And that's why the course that we have, it's actually a fairly extensive course. And just from the nutrition standpoint, it's a 3.5 hour long lecture. So when it comes to weight loss genetics, using genetics, we can identify what kind of dietary interventions will expedite weight loss for the individual, what will not expedite weight loss for them. Again, the fitness regimens which will help for the individual in terms of losing weight, and what won't work for them. But overall, once you factor in the diet and fitness based on the individual's genetic makeup, you can have a true DNA-based weight loss plan which will have efficacy. Now the next part we'll be going over is the dietary components. How many people think coffee is good for them?
You missed the class before.
And how many think it's bad for them?
I'm in the middle.
We're in the middle.
Yeah.
I'll go back.
Good and bad.
Yeah.
There's no answer to it.
Yeah.
It really depends on the genetic
for me.
makeup of the individual. And the missing link between good and bad is a gene called CYP1A2, which is part of the cytochrome P450 complex. So when it comes to caffeine, caffeine inhibits the adenosine pathway. So adenosine normally binds to its receptors, and that leads to dilation of the blood vessels, which drops your blood pressure. What caffeine does is it inhibits the entire pathway, and that leads to vasoconstriction, which increases your blood pressure. So if you're having coffee, and if caffeine is present in your blood for a long time, you will see the adverse effect, whereby there will be vasoconstriction, and that will be increasing your blood pressure. But for some individuals that have a heightened activity of this enzyme, caffeine is rapidly broken down to these constituents: paraxanthine, theobromine, and theophylline. And these constituents are actually really good when it comes to your cardiovascular health. So it's really a balance of how rapidly this enzyme is working in your body to break down caffeine. And that's why there is no universal answer in terms of is caffeine good for you or bad for you. And again, it's really a single nucleotide chain in that gene that determines this. So individuals that have the 1F allele, they have reduced enzymatic activity. In those individuals, caffeine stays in their blood for a long time, and of course, they have a higher risk of hypertension. In contrast, individuals that have the 1A allele, they have very high levels of this enzymatic activity. Caffeine is rapidly broken down to those individual constituents. For these individuals, if they drink coffee, they're reducing their risk of hypertension. So this is actually a summary of data. I think it involved over 10,000 participants. And the take-home message really is, so if someone is a slow metabolizer, if they were to have over 4 cups of coffee a day, their risk of hypertension is going up 3-fold. But again, the opposite is true if you're a fast metabolizer. If you're a fast metabolizer and you're having that much coffee a day, you're reducing it, not increasing the risk. So overall, you have to look at someone's DNA, and then only you can answer if coffee is good for them or bad for them. And same goes for almost every dietary component you can think of. There is no universal answer in terms of it's good for you or bad for you. Along with that, we also cover food intolerances like gluten intolerance, lactose intolerance. So when it comes to lactose sugar is broken down by an enzyme called lactase, which is encoded by the LCT gene. And this gene in itself is regulated by another gene which is called MCM6. So if somebody has increased activity of this gene, that will have a positive feedback on lactase production. And in those individuals, what you have is lactase persistence, because they keep on producing lactase, so they break down lactose effectively and they do not develop lactose intolerance. In contrast, individuals that have the G allele, in those individuals, MCM6 is not as active. So that's why it has a negative impact on lactase production, and those individuals have a higher risk of developing lactose intolerance. At the same time, when we talk about gluten intolerance, it really comes down to the ability of your body or your immune system to present gluten in a form that your immune system is activated. So gluten intolerance is not as straightforward as lactose intolerance. It's something which involves over 15 different genes, and then only you can truly identify if someone has a risk for gluten intolerance or not. So using dietary genetics, we can help identify if someone is at risk of lactose intolerance or gluten intolerance. Is coffee good for them, bad for them? And of course, we hear all the time that sodium is the worst thing for you. But again, not everyone that eats a lot of sodium goes on to develop blood pressure. And again, that boils down to your genetics. Even things like if you're having a lot of starch, what is your risk of developing insulin resistance? There are some people where if they're carrying that high risk genotype, they should effectively be cutting down on starch intake. Along with that, using genetics, we can identify if they have a sweet tooth, if they have a big snacking tendency, and also if consuming whole grains can reduce their risk of developing type 2 diabetes. Because even though we hear in the media that whole grains will cut down your risk of developing type 2 diabetes, it's not true for everyone. It's only for a certain chunk of population. The next segment I'd like to cover briefly is the detoxification. So when it comes to detoxification, of course, the liver is the site for detoxification. All exogenous chemicals, pollutants, medicines, hormones, everything is broken down in the liver. And the enzymatic pathway that plays a role in the process is, of course, the cytochrome P450 complex. So when it comes to the detoxification pathway, it's really a two-step process. In the first step, there is a chemical change which is brought upon, whether it's a hormone, chemical, or whatever is going into the liver. And in phase 2, there is addition of certain chemical groups to it to make it water soluble. And once it's water soluble, it can easily be eliminated through your body by urine, bile, or stool. Again, two examples to illustrate the role of detoxification pathways. So when it comes to cough and cold, none of the over-the-counter cough syrups have shown any clinical efficacy. It's really the biggest load of crap out there. I mean, quite frankly, not a single clinical study can even support they do anything. The only thing that works for your body is codeine. But again, the problem with codeine is, for certain individuals, when codeine goes into their body, it can be rapidly converted to morphine. And that just depends on one enzyme, which is CYP2D6. So individuals that have high levels of CYP2D6 activity, let's say they were to take codeine for cold, cough, whatever, in those individuals there would be a rapid conversion to morphine, and next thing you know, they're showing symptoms of morphine overdose. This is a classical example of pharmacogenetics, that how is your body going to respond to a drug. But again, that's true for over 150 different medications that have been identified. Even things like, for instance, let's say somebody goes to get help for depression, they're prescribed a medication. Again, this is something that happens all the time. They're given a medication, they go home, 6 months later they commit suicide, and the clinician is like, oh, why did that happen? They were on a certain medication. It's because that medication did not work on the individual. That's why in certain markets, especially in the US now, there is a strong push to make pharmacogenetics testing mandatory. In other words, beforehand, just identify what's going to work on the individual and prescribe medications accordingly. You can't take those chances. Yet we do every single day on a global scale because of the underlying cost of genetic testing. And of course, the other side of detoxification is breakdown of hormones. So estrogen by itself is actually a cancerous hormone because it promotes tumor formation, it promotes growth of breast cells. And our body has a very elegant system in place to break it down and eliminate it from our body. But again, our body also has an ability to convert estrogen into cancerous metabolites. And that really depends on few detox enzymes. For instance, if somebody has very high levels of activity of CYP1B1 or CYP3A5, then estrogen can be really converted into cancerous metabolites. And for those individuals, you'll actually have a higher risk of developing breast and ovarian cancer. But at the same time, you have GST enzymes, and their function is to inactivate these cancerous metabolites. So again, by looking at the spectrum of all the different enzymes that play a role in estrogen breakdown, you can identify if somebody has an impairment. And if so, you can truly identify at what level, and you can have a dietary intervention accordingly. So the example I'll be going over today is that of GSDM1. So GSDM1, its main function is to eliminate a lot of these toxic compounds from the body by making them water soluble. But again, when it comes to GSDM1, there are many different forms of the genes that you see on a population level. For some individuals, they have very low levels of activity. Some have very high levels of activity. Now, how does this relate to estrogen metabolism? Now, if someone has very low levels of activity, they are at a high risk of developing estrogen-dependent breast cancer because the cancerous metabolites are not being readily eliminated from their body. What is it that you can do about it? The good thing with the detox enzymes is they are inducible and you can also suppress them. So this is taken from an in vitro study whereby lymphocytes or blood cells were taken from an individual that had low levels of activity of GST enzyme, and when they were given resveratrol, there was a massive induction in activity. So in other words, by looking at someone's DNA, you can identify if they have a GST impairment, and if so, you can actually give them supplements, give them certain herbs which have been shown to boost the activity of the enzyme. So what does it all mean? Of course, using DNA, you identify genetics of estrogen metabolism. You can identify if genes that are involved in clearance of these cancerous metabolites have reduced activity. If so, then you can have a dietary intervention to boost their activity. On the flip side, you can also identify that if genes that are involved in conversion of estrogen to toxic or cancerous metabolites have increased activity. If so, then you can have a dietary intervention to reduce activity. But in a nutshell, by looking at someone's DNA, you can identify where that impairment is, give them a dietary plan based on that genetic impairment to reduce their risk of developing breast and ovarian cancer. So again, just a very simplified case study. So let's say we look at the genetics of an individual, and let's say we look at their GSDM1 activity. The activity is decreased, which means they have reduced conversion of toxic estrogen metabolites into non-toxic forms. So what that means is you would need to boost the activity of this enzyme to make sure you can reduce the risk of them developing breast and ovarian cancer. Similarly, the other example is, let's say if you were to look at CYP1B1, it's an enzyme that converts estrogen into 4-OE1, which is a cancerous form. And if the individual has increased activity of that enzyme, you want to give them a dietary support that can reduce the activity of that enzyme. For the last segment, we are briefly going to cover diabetes. So of course, when it comes to the monetary burden that diabetes has, I mean, it's on another level. I mean, 174 billion, that was in 2007. But now the latest stats are saying that around 45% of the population is pre-diabetic.
Wow.
Pretty much what it means is they're almost there, and they're just waiting to become diabetic. Because you won't go to a doctor if you're pre-diabetic, you're not showing any symptoms. But diabetes in itself is not a single gene disease. Around 13 different genes interplay with each other to mitigate or to put you at risk of developing type 2 diabetes. This is a very simplistic approach in terms of understanding how these genes work with each other, but I think it explains the point well. So let's say there's an individual, they have certain genes they get from their parents which makes them hungry all the time. So now because that's the case, they'll be going and eating a lot, and next thing you know, they become overweight. Now let's say in parallel they have another set of mutations which puts them at risk of insulin resistance. You mix the two together, what happens is they are increasing their risk of developing type 2 diabetes because they're genetically prone to it. Now let's say there is another set of mutation that they're carrying which impacts the activity of the beta islet cells. You put all the mutations together, and next thing you know, the individual goes on to develop type 2 diabetes. So what we have developed is actually an AI or deep learning powered algorithm which factors around 13 different genes of an individual, and looking at the genotype of these different genes, how they're interacting with each other, we can identify if the person is at high risk, moderate risk, or low risk. When we say somebody is at high risk, what it means is that they have around 80 to 90% chance of developing type 2 diabetes. The question that we get all the time is, so let's say if you identify that somebody is at 80 to 90% risk, what does it all mean? Like, what happens then? Well, what you got to do is you have to take a multi-pronged approach to reduce their disease risk. So how we talked about diet, nutrition, and fitness, first of all, you got to have a plan for the individual based on their DNA, a weight loss plan, again, based on their DNA. Moreover, we do a fairly extensive analysis of the complete detoxification pathway, which tells you what the adverse effects are, and accordingly, you can do modulation of detoxification pathways. And the other key point is, once somebody, let's say somebody is highly obese, the moment they know they are at 90% risk of type 2 diabetes, once you give them a health plan, they're very likely to adhere to it, versus them not knowing the information that they are at 90% risk. So final thoughts, when you provide a non-DNA based health plan, there are three outcomes. Either it's not going to work, it'll have slight efficacy, or it'll have very potent efficacy. But when you're providing a DNA based personalized health plan, because it's catered to the biology of the individual, the only outcome that you're likely to see is potent efficacy. Now again, I'll just go over a little bit about Anantlife. So after I stepped out of the academic world, that's when Anantlife came about. And our mission is really to provide low-cost, affordable, next-generation sequencing-based genetic kits, which are clinical utility. And the way how it all works is through a saliva sample. So the patient gets a saliva sample. There is DNA that we extract from the cells. We sequence out the entire DNA. We compare it to our reference database that we have developed, and then we have certain deep learning algorithms that we use to identify the risk of the individual. What makes us unique is, I guess the biggest of all is that it's a clinical genetic test. It's not a consumer-based genetic test, because the moment you're offering a consumer-based genetic test, you can have 30% accuracy and it's all okay. And perhaps the biggest example is 23andMe. If you were to take the data of 23andMe to a healthcare provider, they would be like, if you just throw it in the garbage. Because they're in the business of accumulating data and accumulating data. That's where it ends for them. And the biggest differentiator is, the moment you talk about a genetic test that has clinical utility, always look for this, which is the next generation sequencing platform. What it really means is, for any gene that you're looking at, you have to read the entire thing at least 50 times to ensure you have greater than 99.5% accuracy. And part of the reason why we have to do it is because of the different array of genetic tests that we offer. For instance, we have a genetic test that can identify if someone is at risk of developing breast and ovarian cancer, prostate cancer, even things like, are they at risk of developing aggressive prostate cancer? Because if that's the case, the treatment modality has to be different. You cannot take the normal wait and watch approach, which is taken with prostate cancer. Similarly, colorectal genetic test, autoimmunity, and brain disorders, where we are looking at things like Alzheimer's, Parkinson's, and Huntington's. And imagine you look at someone's risk for Huntington's. If they're at risk, what it means is the chances of them developing the disease is 100%.
Wow.
So if you are off there, let's say you tell somebody they're not at risk at all and they go on to develop Huntington's, it's a massive lawsuit. And that's where the biggest differentiator happens. The genetic testing providers that are giving clinical genetic testing, they will never be selling it directly to consumers. Because as a consumer, let's say you have no idea about DNA, you get this report, what do you get out of it? Yeah.
So if we're practicing, are we the ones providing?
So the ones that you guys can provide is the health and wellness genetic test. We were also the first ones in the world to develop a genetic test to identify if you're prone to adverse reactions from cannabis. Because as much as we talk about the medical side of cannabis, and with any medication, you'll have a subset of population where it's going to work, subset of population where it's not going to work, and subset of population which will have a lot of adverse effects. And that's what we can identify through a cannabis genetic test. And of course, pharmacogenetic tests, where you can look at the response to different medications of the individual. There is one more test that we offer, which is the neurogenetic test, and it's about looking at the genetic risk of developing autism. And it's more so looking at a bunch of different pathways, identifying where the impairment is and what can be done from a dietary and nutritional standpoint. The same test also looks at risk of addiction, depression, schizophrenia, OCD, and so on and so forth, along with identifying what the intervention should be for the individual. And, well, this is not relevant for this audience. So just to briefly go over the health and wellness genetic test. So when it comes to the health and wellness genetic test, it's more of a high-level summary of what's going on in someone's body. In terms of what it's covering, so we are looking at the complete detoxification genetic profile, estrogen metabolism. Let's say if it's a male patient or a male client, risk of testosterone deficiency, vitamin and mineral deficiency for all the vitamins that were listed initially, risk of developing high blood sugar, cholesterol, triglycerides, again, risk of developing gluten and lactose intolerance, their fitness genetics, weight loss genetics, and alcohol dependence. And of course, I mean, ever since we had the association with IHN, we always have been offering a 50% discount on the account setup fee. So the way how it works is, once the provider sets up their account, we actually provide them with 4 kits to get started. And normally what we charge healthcare providers is $320 for each and every test. But as IHN members, alumni, you guys get 10% off on that. And we only bill for the test once we get the sample. And of course, the account setup fee is a one-time fee. And as you run out of kits, we send you more kits at no extra cost. And as I said, it's a saliva-based genetic test, and the good thing about saliva is it's stable at room temperature for 6 months. So from a logistics standpoint, it's not about you get a sample, you have to stick it in the fridge. You just have it sit there at room temperature. And of course, coming to the course. So last year was the first time we started off with this course, which is Understanding Genetics for Improving Health Outcomes. And the real purpose of this course is not to make sure that you memorize what DNA is, that you memorize what a certain gene is. It's more about how can you take all the tools and use them to do an interpretation of a genetic report and provide specific guidelines for the patient. And that's the sole purpose of the course. It's not a typical university course where you guys will have to, let's say, go through, oh, did you memorize this, did you memorize that? We have a midterm and a final, and that's about doing case studies. So you're actually given reports, and you do an interpretation of the report based on what you have learned in the class.
Is there a lot of homework in that class?
There's no homework, just
No?
just a midterm and a final.
Midterm and final.
It's take home, both of them. Yeah.
How many weeks is it?
8.
8 weeks.
Yeah. Yeah, and that's it. And of course, I got brochures for our health and wellness genetic test and the account set of forms, so feel free to get your hands on them. Any questions? Yeah.
I have questions, but you didn't talk about it, but it's something related to telomeres.
Yeah.
So we've just been doing our own research, but what's your feed on how can we lengthen and strengthen our telomeres? Because like in our research, we found that it helps to extend life.
So, I mean, at the end of the day, it's, so when it comes to telomere length, it is going this way.
Yeah.
So, I mean, if that was not the case, we should potentially be able to extend our lifespan to infinity. But over time, as cells are dividing, telomere length is going to be becoming shorter. You can reduce the rate of acceleration, but at the same time, it's not something that you can eliminate.
Yeah.
Yeah.
But can you tell us some things of how we could reduce the rate of the shortening of the telomeres?
So again, when it comes to that, I mean, there have been some claims.
Yeah.
But again, I don't think there is sufficient backing of scientific data to support those claims.
Okay.
Because again, when it comes to telomere length, it's something that every mammal or every organism deals with that has chromosomes.
Yeah.
And but I mean, I won't comment much on it simply because, again, from a scientific standpoint, there hasn't been much validation in terms of which approaches work the best. For instance, certain antioxidants are supposedly said that they can help reduce telomere length. But again, as I said, every time your cell is dividing, telomeres are shortening. You don't want to impact your cell division because the moment you impact that, you're increasing your risk of developing cancer. It's about, my take is it's more about making sure that as cells are dividing, you don't introduce any new mutations in the cell. Because as you start to do that, you're increasing your risk of developing a lot of diseases. And that brings the whole tumor suppressor genes into question.
Okay.
As part of the testing, do you see the size of an individual's telomeres?
Yeah.
And you understand where they are in the age spectrum?
Of course, and that's the biological age of the individual.
Yeah. That's the fascinating
Yeah.
part. Yeah.
Wow.
Yeah.
Because even the rate at which your telomere shorten is governed by the genes that are present in those chromosomes. Because you got to remember, when cells are dividing, the entire process is governed by your DNA. So I mean, so it's a very inquisitive problem in itself. Imagine something is shortening, but at the same time, the rate at which it's shortening is governed by the same thing itself.
What does the market look like right now? I think this is a two-part question. The DNA market in general, these tests are done once typically. Yeah. And then a client won't necessarily do another test.
So the way how we approach it is, so let's say somebody starts up with health and wellness. There are certain red flags that are identified in there. So let's say based on their GSTM1 activity, they are at a risk of developing breast and ovarian cancer. Now, maybe based on their family history, they may want to explore the breast and ovarian cancer genetic test.
Got it.
Yeah.
Okay.
So think of the health and wellness as your first assessment that you have with your healthcare provider.
Yeah. And how would you see, so 4 kits come for $288 is what I saw there?
So 4 kits, so there's a one-time account set of fee.
$99.
Yeah.
And then 4 kits.
And so you get started with 4 kits, and it's when you ship the kit back to us. So one test is going to be $288.
I got it.
Exactly.
So with the $99 we get the 4 kits.
That's right, and that's a
distribute.
one-time fee. Yeah.
So markup then. So if I'm selling this to a customer,
Yeah.
what's the typical market rate to do one of these
So
tests?
with health and wellness genetic tests, for instance, it's really the report is around 20-25 pages, and almost every section is one consultation. So let's say vitamin and mineral section is going to be one consultation.
You're saying you're building consultations out of it. It's not like you're charging them for the test.
So overall, what especially a lot of IHN alumni, what they do is a lot of them actually charge $100 markup on the test. But what others do is, depending on the clientele they have, is they're going to offer a much bigger package.
Sure.
Test with, let's say, 5 or 6 consultations along with it.
Yeah.
And that's where they increase the ticket of the entire transaction.
Okay.
But again, the other side to that is private clinics, private health clinics.
Yeah.
There, the market is just on another level where I look at that, I'm like, how do they even do it? Like, they can go to thousands of dollars. I mean, especially with the private clinics.
Wow.
Yeah.
I have a question about epigenetics.
Yeah.
You will touch base on that? With
That's in the course. I mean, not today. But I mean, yeah, that's something to go over in the course. Yeah. And at the same time, so when we talk about the detox pathways, right? So these components, when they're inducing or repressing a gene, that's all happening through epigenetics because the DNA itself is not being modified. So when you talk about epigenetics, it's really when a part of the gene is highly methylated, a lot of methyl groups are present, then the activity of that gene will be reduced. If you get rid of those methyl groups, though it's the same DNA, you'll have a lot more activity. And that activity happens at the level of converting DNA to RNA. That's the rate limiting step. And that's where the dietary intervention makes a difference.
Okay.
Is that where you would see more blood samples telling you, giving you indications for epigenetics?
No.
No?
No.
Okay.
What about like the test? Will you teach us how to read it? And like I can when I get the test, will I be able to read it and recommend like through the
That's right.
the course, or will like when I apply to?
So I mean, let's say somebody said, I mean, of course, it's not like everyone who's doing our testing is taking the course, but definitely we do provide like a guide to interpretation and all that. The advantage that people who take the course, what they tell us is that now they have a complete understanding of everything that they're reading, saying, and telling the patients. That's where it makes a huge difference. That now they almost have the freedom to some extent, go beyond what's even being said in the guide to interpretation, because they understand how all the ABCs are working and lining up.
Yeah.
But still, like I don't understand how as a practitioner will help me to know these information. As if I know like how to detox something, it like let's say like I have the mutated form of methylation.
Yes.
Let's say, why I don't give the patient the already the folinic acid, let's say.
Because I mean,
without
because when it comes to detox pathways, a component that will activate one can repress the other. And repressing the other can increase the risk of cancer as well. So by blindly offering, or maybe more like giving a generic solution, what you're doing is you're indirectly playing around along with the health of the individual, where you may see adverse effects with a different pathway. And that's where it's about understanding how the different pathways are working along with each other, and then you come up with a plan based on what the individual, because let's say we talk about 2 different pathways, and there is the same component that can induce both the pathways. But now let's say for the second pathway there is another adverse effect which is associated, then you don't want to use that component. You want to use another dietary component which is specific to first pathway.
Does that happen with MTHFR?
I mean, not with MTHFR, like overall with
That's
detox
what I
pathways
mean.
that happens really commonly.
A lot.
You can do that, like you need this with medication, but do you need this with supplements? Do you need this with
So when it comes to the
nutrition?
induction and
Like I understand with medication.
No,
because,
I'm not talking medication. I'm talking about the detox pathway. So when it comes to the induction and suppression, it's not medications. It's herbal supplements, herbs that have been shown to induce or repress.
Herbs are like medication, but it's safer.
Yeah.
But in the same time, I mean supplements, like as
Well, there was
Joe
the
said,
quercetin example.
Yeah, like I
As
don't
supplement.
understand like supplements and nutrients. Do I need to know?
I mean, of
what's
course.
the side effects.
Absolutely. Because again, so that's the thing. I mean, so I think the health and wellness test is by far the one that's the most relevant to the crowd here, because everything that's being covered is all about your diet, nutritional support, and supplements.
Okay.
Yeah.
What about if we wanted to get tested? Do you have a special offer for IHN students for that?
No.
No?
We don't.
Do you have like a sample results? Of,
Yeah. Well, of course. Well, I mean, there was actually an email in the brochure. Just grab one. I mean, shoot an email to that address and somebody can send you a sample report.
Okay.
Does your collected information go up on any kind of database that can be accessed? don't mean by us, but by, let's say, any policing authority or whatever?
No. And at the same time, with every test that we do, there is a consent form which actually says, if you want us to completely destroy your information after the test, you can just put a check mark there, and then we have to delete everything.
Good.
Yeah.
As a practitioner, wouldn't I be able to offer my customer, like if I send you a sample, I just won't put their name on it. I can anonymize by sending to you, calling them Jane Doe or John Doe, and then you return the sample back to me, or the results back, and I know it's Jane or John Doe. So I've kept anonymity.
Yeah, you can even do that.
Works,
That's,
right?
so at the end of the day, I mean, every sample is barcoded,
Yeah.
and that's what our system works on. It does not work on names, it just works on that barcode.
Yeah. Awesome.
Do you have this course every year or only this year you will have? So
No, it's going to be every year.
every year,
Yeah.
yeah.
So if, with this package, can I do a test on myself though right now?
I mean, of course, yeah.
Yeah, okay.
I mean, you can use the kit on anyone who wants to.
Yeah, because that's what I want to do.
It's all about you, isn't it?
Well, first heal myself and then, you heal others, right? Yeah. Okay, perfect. So I just need this and,
Yeah.
okay.
Thank you.
Thank you so much.
Oh, no problem.